Hello everyone, welcome to today's webinar. This is a webinar collaboration between Azure Biosystems and Apical Scientific Malaysia, where we talk about Elisa on Ao Absorbance Microplate Reader. So this is Ao Absorbance Microplate Reader, and we will talk about its basics and application that you can done on it. I'm Leng, the Regional Field Application Scientist of Azure Biosystems, and let us start with a little bit of introduction to the company. So Azure Biosystems, we are a team of highly experienced scientists, engineers, and entrepreneurs dedicated to accelerating your science. So all our products that we produce and manufacture combine smart and simple workflows that gives you high performance and affordability. And so you can have the confidence in your data to move forward quickly and also flexible capabilities to go whenever the data takes you and what kind of signaling pathway do you want to do next. So our company founded in United States, California, very near to Silicon Valley. Our company is founded by Onno Shifji. He's our CEO and chairman. He's a venture investor. And Dr. Diping Chair, our executive vice president of R&D. So if you look at the Dr. Diping Chair past experience, he has a lot of experience in developing protein imaging system in Alpha Innotech and Illumina. Let me brief you through a timeline of the development of Azure Biosystems. The company is fairly new. It's founded in 2013. All the core members are coming from Alpha Innotech. So if you've been doing chemiluminescent or Western blot imaging or gel imaging, you'll be very familiar with this brand. I'll say it's a very old brand, but it has been on the market for a very long time. But nowadays, it's bought over become protein simple. So our company basically founded the core members are coming from Alpha Innotech, and we have a few key instruments in our company portfolio. The first one is Ao Absorbance Microplate Reader, which we introduced in 2016. We also have Sapphire Biomolecular Imager, which is the industry first hybrid CCD imaging system and also laser scanning system. It's still our highest spec instrument until today. And we also have Azure imaging system, which is focusing on gel and Western blot imaging. And of course, we have also a genomic qPCR system as well. But today, we will be focusing on Ao Absorbance Microplate Reader. Okay, so let me tell you what's content of today's session. Firstly, we will talk about the basics of enzyme-linked immunosorbent assay. The short form will be ELISA. Then we will look at the application example you can do with an Ao Absorbance Microplate Reader, where I will be explaining one of the ELISA application node that we have. And also I'll be briefly go through two publications that done with Ao Absorbance Microplate Reader to let you get a grasp of how the data looks like in a very fundamental form and also in the advanced format. Okay, so first, let us look at the basics or the principles behind ELISA. Enzyme-linked immunosorbent assay. So this is a very widely used biology research method to quantify protein or any other antigen in an unknown solution. So here we have the word immuno. Immuno means being anything antibody related. So we're talking about protein interaction here. So it can be protein, it can be protein DNA, and all other stuff. But the fundamental thing is antibody based. Okay, so these immunoassays often rely on the sensitivity and the specific detection of the antigen by the antibodies itself, okay? So which need to be repeatable and relatively quick to perform. So ELISA, as we know, the data need to be repeatable, okay? You need to have at least 3 replicates and to know that the assays are functioning good. And the result must be relatively quick for you to generate it. So it shouldn't take a very, very long time because it all boils down to the sensitivity and the specificity of the ELISA kit itself. So in order to achieve this kind of repeatability and also convenience and quick, these the two functions of the antibody must be, I'll say, optimized. So these two functions are capture and detection. So capture here, what we means it will be antibody with like affinity, high affinity or low affinity, antigen selectivity, and the specificity. So for example, antibodies with very high affinity and selectivity for their antigens are very good for pulling and holding the antigens out of any mixed solution. So when you wash this, I'll say washing step, those unbind proteins or other stuff will be washed away. Okay, so antibodies like these will be very good to use to prop onto the surface of, let's say, a plate or any other thing, then allowing you to to incubate it with your unknown samples. Okay, so the example will be applied in ELISA kit. So such antibodies are usually monoclonal, okay, because they have high degree of specificity with the potential of detecting very low amount of sample, the sensitivity wise. Okay, conversely, you also can have antibody that has very quick binding rate, but the affinity will be lesser. It will be very good to work on sample that already been immobilized. For example, like IHC or ICC, where you have your sample on the glass slide, then you probe with your primary antibody. Okay, so this is how people use the antibody, each of their, I'll say, characteristic to design the entire assay. So for ELISA context, we use those high affinity and selectivity and specificity for ELISA to probe them and fix them onto the plate. And we also have detection type, which will be different type of incubation that giving you different range of detection and whether it will be cheaper or whether it will be more lengthy. Then the next slide, I'll be focusing on how the detection works in general immunoassays. So we have direct, indirect, and sandwich. Okay, let us look at the direct detection type. Direct detection type here, so the example will be you have antigen probe on the surface, and then you have your detection antibody, which is conjugated with any kind of thing that gives you signal, can be HRP, can be also fluorophores, depending on what kind of result, and also others that give you colorimetric changes as well. So this type of detection, direct detection, is the most rapid protocol of all. Is a bit less stringent because it's just one bind. So the specificity will not be that good. So it's not requiring ultimate accuracy, okay, for this direct type of detection type. For indirect detection type, where we have antigen, we bound with primary detection antibody, secondary antibody, and we have a secondary antibody that conjugated with your signal molecules. This kind of indirect detection type, it improves upon the specificity of the direct detection. So because you introduce a secondary antibody. So this also adds benefits for allowing signal amplification, which is very important for low abundance samples. Okay, right. So you have the indirect detection type, which gives you higher sensitivity and it amplify the signal for and good for those low abundance samples. Okay, right. Then third one will be a much more interesting one. We call it sandwich detection type. Sandwich immunoassay, these, okay, either be sandwich direct or sandwich indirect, they provide the greatest level of sensitivity and detection because it's sandwich, it's a lot more. And the capture antibody will be probe or immobilized onto a specific surface, just like how we see in the ELISA, and then you expose it with your antigen, okay, with your sample. Then this can then wash off, okay, you wash off the sample to remove those unbound or weakly bound product. Then you incubate with a detection antibody, or the detection antibody can be either conjugated conjugated with your signal for I'll say dyes, or you have another secondary antibody that also probe with the conjugated with the dyes itself. So this time of we call it sandwich direct and sandwich indirect. This is I think much more familiar and applicable in ELISA plate. That is what we expected in ELISA plate mostly. Okay, so this are the detection, as I said, it gives you the highest sensitivity and detection. Okay, so you can detect a very low amount of I'll say uh, abundance of sample, and the detection will be much easier. So it's easier for you to plot the standard curve. Okay, and then it can be, but the downside will be, okay, of all these steps is definitely increase in the assay length. So it takes a longer time because you have to probe with your sample, probe in the primary antibody and probe in the secondary antibody. So it takes way longer time. And also the difficulty to match the antibodies, okay, from here to here and from here to here, that bind to the same target efficiently and without interfering each other's. Okay, but for end user, like most of you and us, we don't need to worry about this part because this will be definitely optimized by the manufacturer side. Okay, so let us look at how this is applicable for ELISA setup. Okay, so there are 4 ways. Okay, ELISA can be set up. Okay, so the first thing definitely is direct, indirect, and sandwich, like I said before, which normally we see in ELISA kits. And there's also the final one where we call it competitive or inhibition kind of setup. Okay, so competitive or inhibition ELISA, the setup can be seen, it can be either direct or indirect or sandwich, it doesn't matter. But instead of the antibody being labeled, okay, like here, you actually label the antigen. So the labeled antigen will be added after you probe the plate with the unknown sample. So you will see how these how much unlabeled antigen in the sample compete with the labeled antigens. So these will be provided in the kit itself. Okay, so unlike these three types of setup, so here these three direct, indirect, and sandwich, where you have higher signal means you have more antigen, right? But for competitive and inhibition, when you have higher signal, means you have lesser competition, less antigen in the unknown. So this is reverse, okay? Let us look at the principles of each of these steps, the ELISA steps, okay? So this is a sandwich ELISA. All right, because you can see here, the specific antibodies are bound to the bottom of the well, what you expect in the ELISA plate. So this will call the plate coating step. You can it's either be available when you purchase it, or you have to do it by yourself. It's up to the kit. Okay, so you follow the manual, then so you block. Okay, to remove the any unspecified finding, then you add the antigen containing samples. This will be your unknown sample. So, okay, let us look at the steps. Probing, then you have adding your unknown samples into the wells, okay, incubate it, then you wash away those unbound samples. So, assuming that you have your antigen is binding to the antibody at the bottom, then you add on another antibody over here, okay? So this will be a second antibody, will be for the detection one, okay? Then this is a enzyme link secondary antibody. Okay, so antibody linked to enzyme is added to detect the antigen, right? Then incubate and wash away those excessive ones. Then add a substrate to convert. The enzyme will convert the substrate to a signal, either be colorimetric, color be fluorescence. Then you subject the plate to a microplate reader to read for, let's say for colorimetric signal will be absorbance reader to determine the presence and also quantity of the antigen. So the two things that you can get from this result will be yes or no, is there antigen or presence or no, and how much presence and quantity, two things. Okay, right. In the usual form, in the ELISA, usually it performs in 96 well plate format because you need to test a lot of conditions like grouping, and you also need to repeat the samples at once. Like say, I highly recommend users to do at least a triplicate, okay, of your samples or anything, including the standard curve as well, to ensure the entire assay. Assay means the one run of the ELISA is valid. Okay, so because you need to make sure your positive and negative control are working fine, so the entire unknown sample data is considered true. Okay, so in terms of commercial kits that are available on the market, they are usually optimized in antibody pairing and also cross reactivity. And also they will usually comes with positive control sample for you, and where they already optimize the signal linearity, means the signal that you get from the ELISA will be directly proportional to the amount of antigen that you get, or inversely depends on the kind of the setup. It will be competitive or non-competitive. And of course, it will also state the optimal range of the ELISA kit itself. It will tells you what is the optimal range of operation of the sample that they offer, they can do within the kit. So unfortunately, if you have sample that is like way lesser, I'll say antigen presence, the quantity, you would need to find a more sensitive kit to do so. Okay, right. So all this is just part of the criteria that you need to consider when you select your ELISA kit. Application wise, it's a very basic thing. We're talking about ELISA. So you can detect anything basically as long as the antibody able to detect it. So it can be applied in food industry to detect food allergen, authenticity of the food. Let's say you have let's say product is a lamb product, then you have to convert whether it's lamb or not. Okay, and contamination check, vaccine development, like you can test the presence of antibodies, diagnostics, pathogen infection in humans or plant, and cancer marker, pregnancy tests like hormone detection, and more and more. So there's no limitation in terms of the ELISA application. As long as antibody can detect it, that's it. Okay, so the limitation will be whether this is an unknown antigen. Therefore, you do not have existing antibody that has been optimized and able to detect and know that to detect this antigen reliably. Okay, so there's no limitation of that. Right, next, let us look at the application examples with Ao Absorbance Microplate Reader. Absorbance reader. Okay, so it's a very basic microplate reader. What it does is you use a lamp, okay, and a filter to read the absorbance at a specific wavelength. So you have here. Then here is a detector. So this is the this kind of setup is at a maximum sensitivity and gives you the best read speed for kinetic assays. And it's very flexible because you just need to change a filter, then you can read a lot of type of assay data. And this kind of absorbance microplate reader is very cost effective as well. This is, I'll say, the most fundamental and basic piece of equipment that you should have in every single lab. Right, so here's the Ao Absorbance Microplate Reader. Okay, it has a very intuitive large format touchscreen interface, so you can run your essay and also view your analysis. And here's the part we have a motorized door to easy access to your 96 well plate. And of course, this machine allows analysis for pre-processing, for dual mode reading, plus or minus interpretation with controls, concentration determination, curve fitting, kinetic measurement analysis, and curves. And this Absorbance Microplate Reader also have built-in shaker with multiple speed selection. Okay, it has single and dual channel wavelength detection mode, which means you have one that want to detect and also one with assigned with reference a channel. Okay, and in terms of filter, it has 8 precision filter wheels, each of them. Okay, with 5 standard filters that covering most of the assays of the market, and also 3 user selected filters from 340 to 750 nanometer wavelength. So you have 5 fixed plus 3 user chosen filters. So we call it the rainbow assay and your fingertips because it's such a basic equipment. So you have a filter for every single assay. So the 5 standard filter will be 405, 450, 492, 595, and 630 nanometer wavelength for most of the ELISA, Bradford, and Lowry assays. Okay, so ELISA is ELISA. For ELISA application, Bradford and Rowley are most for protein concentration determination that you can use it for further to determine the protein concentration in your island sample. So you can do the rest, quantifying the protein and do the rest of the techniques such as like Western blotting. Okay, and also you can choose 3 filters out of the range selections. Then you have a total 8 filters for wide coverage. And of course, as a machine, okay, equipment, a piece of equipment, it must be reliably precise, accurate, and give you linearity in signal. So you know and you safely know that the machine is reliable. Okay, what you get and what it is. Okay, so I will not go into details like read speed and accuracy because these are basically the basic thing that an equipment should be complied to. Okay, right. I'll focusing on the application over here. So here's the example of the AC that you can read with each of these filters. Okay, so we call it a rainbow AC because it covers most of these wavelength. So you have 340 like NAD and NADPH conversion, alkaline phosphatase detection, or horse radix peroxidase detection, or concentration, and MTT and XTT assay and MTA assay, most for cell viability assays. BCA, protein concentration determination. Okay, L-ascorbic BCA also protein. Okay, and reference normally at 630 nanometer wavelength and more and more. So you can basically select the 3 optional, not types optional, the user selected filters from here, and we have more as well, which are didn't display over here. So basically, these range of filters, it can cover both of your ELISA needs. Okay, nucleic acid quantification. Yes, you can do nucleic acid quantification using ELISA plate reader. Sorry, microplate absorbance microplate reader. Protein quantification, enzyme kinetics, and also any other calorimetric assays. Okay, so calorimetric is such a basic things, but it fulfills the needs and give you very fundamental and crucial information. So here's the UI that you will see on the Ao Absorbance Microplate Reader. Here as you can see, we have dual reading mode. You have the one that is set to read, and also the reference one. Here we say no, there's no. And you also have shakes setting, interpretation, layout will be in 96 format, calculation, pre-processing, or kinetic measurement that you want to show, like export it in a graph format that readily available, and also the quality control. So here's how it looks like in the play layout. You can freely assign the wells using touchscreen. It's just click and drag and drop. That's it. With no negative control, blank. This is standard. Each of them you can assign with the, I'll say the units and the concentration, and you save it as a template. And QC unknown control and also blank. Sorry, blank means empty wells. Okay, so you can just save these play layout in your microplate reader. So here I will showcase to you what is the kinetic parameters that you have. Sorry, the calculation parameter pre-processing that you can have in this absorbance microplate reader. So these basically cover most of the calculation. You have minus, again plus, dividing. So it's based on the measurement wavelength and how it's analyzed according to the reference wavelength. So this is what you call pre-processing setting. This machine also can do kinetic parameters. It generates different kind of a graph that allowing to you can also or without disturbing the raw data. You do have the raw data available as well, but you also can export it in terms of the kinetic parameter graph. So we have average rate, okay, maximum rate of reaction, okay, and maximum time to reach a maximum rate, time to change of the rate, maximum absorbance of each individual wells, and also time to reach a maximum absorbance. So this is how it looks like in terms of the kinetic parameters setting. In terms of application notes, we have 3 at the moment. First will be performing an ELISA with the Ao Absorbance Microplate Reader. Here it listed out all the basic things, the introduction and the principles and the general methodology, and then how you analyze the data and how the data should looks like. Uh, then we have the one that we use with this calorimetric assay, which is Bradford assay. Like I said before, it's meant for total protein quantification. Here's an application to showcase to you how to set it up, again, or what kind of kit that you use, and then and also what's the data you was expected to see. Okay. And next will be more relevant to today's topic, which is measuring the histamine level in food samples. So this is just one of the ways to demonstrate the broad application and flexibility of an Absorbance Microplate Reader. So our next slide will be going to details of these application note. Okay, histamine enzyme immunoassay kit. So this is a kit, okay, well, the kit contained a 96 well plate pre-coated with a monoclonal histamine antibody. So you have a plate with the antibody coated on top. You also have histamine conjugate, histamine standard to plot your standard curve, substrate to generate a signal, colorimetric signal, PBS, and also wash buffer. Okay, so the reason why we have this histamine level, testing histamine level in food sample, because as you know, histamine is a mediator for inflammatory, responses. That's closely related for initial phase of anaphylaxis, gastric acid regulation, blood vessel permeability, and brain function, and blah blah blah. So the histamine actually they do appear naturally in food. So if you have food that is contaminated, I'll not say contaminated, but it's a high levels of histamine, it will actually will be very bad for those people who have reduced diamine oxidase activity. So diamine oxidase means people who is an enzyme to they degrade or convert the histamine. So these people will be suffering for histamine toxicity. So this will be quite bad. This is one of the, I'll say the QC part of food industry where they test the histamine level in food. Okay. So we have the food samples here. We have dilution series of red wine, apple juice, blueberries, spinach, coffee, and walnuts. Okay, so let us look at how's the basic the principle in diagram. So this is a competitive or inhibitory ELISA. So here we have an antihistamine antibody pre-coated on the surface. Okay, then you introduce your sample. Then you expect the histamine from the food sample will be combined and bind to the antihistamine antibody that available on the plate itself. Okay, then you wash it off. Then you put or introduce the competition here, which is the HRP labeled histamine conjugate that is available from the kit. Okay, right. So after this HRP labeled histamine conjugate will be competing with this histamine from your food sample earlier. Okay, so if let's say you have a lesser histamine, of course, more of them will be binding to the antibody, and then of course you get higher signal. So after the incubation, okay, then you added the substrate wash off. You added the substrate to uh I'll say let the HRP enzyme convert it to a signal. Okay, then produce color changes with 1N-hydrochloric acid. Then you read it at 450 nanometer wavelength, and then substrate reference read at 630 nanometer wavelength with Azure Ao Microplate Reader. Okay, right. So this is how it looks like. It's a competitive ELISA. The result that you will see will be over here. Okay, so this is the pre-programmed protocol. We have two dual mode over here, the reading at 450 and reference wavelength at 630, and no pre-processing and kinetics. It's just a basic things. So these are play layout for how they dilute the samples and everything and all kind of dilutions. And then setting of the plate, as you can see, we have this standard curve setting over here. This is they do it in duplicate, if I'm not mistaken. And then they do set the standard concentration for each of these well. Okay, so you can calculate the standard curve and also giving you the data accurately, and so calculate your unknown. So the data over here is, so we have the raw data and we also have the analysis data. So the most important thing in your ELISA, basically you need to look at your standard curve. Are the standard curve, sorry, not here. So other standard curve are giving you a very linear, not to say linear, but very accurate kind of accuracy. So if they are working good and your blank is indeed blank, means there's no contamination in your master mix, then you can safely say that the entire assay is true. Okay, it's true, so you can trust that the results are true. So then, of course, we have this analysis curve, which is, yes, it is a standard plot. It's not a it's not a linear curve because it's a competitive. So you do see this kind of curve in competition in the competition kind of ELISA. Okay, so you can see that how they plot the data from here to here. Okay, so this is just showcase to you how the very basic Ao Absorbance Microplate Reader can give you such a I'll say a fundamental but very important data. Right, so next let us move on to publication. I think this is, some of you might be more interested in it. Okay, the first thing, we have this kind of this publication from Antibiotics, published with MDPI. Impact factor is around 4.6 plus. So where they it's a group of researchers from Thailand. They want to detect the colistin in and other antibiotic resistance in these SI46 multi-drug resistance bacteria. Okay, right. So let us look at the aim of the study. So to try to profile the antibiotic resistance, and also hoping and trying to find the resistant genes from these multi-drug resistant bacteria. So here's a method, a simplified methodology over here. We have isolate. Of course, firstly they need to isolate the bacteria from human sample. Okay, then they subject the sample to the bacteria to antibiotic susceptibility testing with this diffusion and also microdilution. So the microdilution will be the part that they use Ao Absorbance Microplate Reader. Okay, after they get the results saying that, okay, so it might be having a very severe drug resistance, they proceed to do whole genome sequencing, identify the bacteria, and then phylogenetics analysis, comparative pan-genome and pairwise sleep analysis, and more. So they want to get more data from this kind of bacteria. Okay. So this kind of profiling is very important for, I'll say, in most of our healthcare, because we do have researchers, I'll say, for in Malaysia, I think it's IMR. They do profile this kind of a bacteria to understand what kind of evolution and alert if we do see some kind of very severe and a spread out of a severe levels of multi-drug resistant bacteria. So especially when we talk about colistin, because colistin is a last resort antibiotic we have right now in medicine. Okay, so if let's say a patient is completely, the bacteria is completely immune, receives colistin, there's basically no rescue for the patient. Okay, so it's very important for profiling. Okay, so here I want to showcase to you how the researcher do this microdilution method for colistin resistance. Okay, this is a result. I just want to show to you how it usually looks like on the plate itself. Of course, firstly they culture the bacteria, dilute it in the broth, and then they mix it with the colistin in twofold dilution, where they want to get the minimum inhibitory concentration and also check the cell viability using MTT-based staining. Okay, so the MTT-based staining is very simple. So if it's alive, it will be turning to purple. If it's not alive, means completely dead. It will be just clear. Okay, so the absorbance of the Felmason the dissolution is detected at 570 nanometer wavelength using Absorbance Microplate Reader. Okay, and then we also have other kind of analysis, but I will not be explaining in the details over here. And most importantly, you can observe in naked eyes or under naked eyes whether the cells are viable. So here, when they are viable, means the color will be changed from yellow to purple. So the purple one means the bacteria are still alive, even though they still have they mixed with these colistin at different dilution. And you do see that the minimum dilution, minimum inhibitory concentration of colistin will be 512 microgram per ml. Okay, so this is a very straightforward result to showcase to you what is the MIC of these multi-drug resistance bacteria. Okay, of course, we have the positive and negative control over here to showcase to you the entire assay is valid. Okay, therefore, they can move on with the rest of the study. So the reason I picked this publication because you can see, visualize the plate, the color changes here. Okay, so next, let us move on to a higher tier of publication, which is from size of the total environment. In fact, factor is 7.9, if I'm not mistaken. It's quite high, where this group of researchers trying to understand whether the manganese oxidizing bacteria, they can transform the cylindrones spermacine, is a kind of toxin that available in wastewater, and transform it, and whether the transformed product can able to reduce human liver cell toxicity. So the aim of the study is to explore the CYN clearance, the toxin clearance capability in a group of manganese oxidizing bacteria. Then they select the best one, of course, those studying the CYN transformatic job where they detect and identify the transformation product, and also test these transformation product toxicity against human liver cell, because the toxin itself, the CYN, is act directly to human liver cell. That's why it's very important to do these kind of studies. In terms of methodology, of course, firstly, you need to culture the manganese oxidizing bacteria. Okay, then they select the best one. Okay, the best bacteria that are able to transform the CYN, and then they analyze and identify the transformation product. Then they do cytotoxicity assessment of the CYN transformation product on human liver cells. Of course, here will be comparative between the CYN original toxin and also the transformed product. Then they look at the removal rate and proceed with their data analysis and statistical analysis. So let's look at the cytotoxicity assessment part. Firstly, they selected two types of human liver cell. Okay, HEPA G2 and HEPA RG cells. These were actually corresponding to different type of receptor, if I'm not mistaken. So they seeded it into a 96 well plate cell culture microplate. Then they incubate it with a media containing the transformation product. So the treated group will be the transformation product, and you have the positive control means treated with the original toxin, which is CIYN. Okay, then the whole exposure will be lasted for 24 hours. Then the test the cell viability of these human liver cells with XTT assay. This is a similar MTT without lysing the cell, okay, the liver cell, and also neutral rate assay. So these two assays, even though they are both cell viability assays, they are actually act differently. The principles are a bit different. XCT is used to measure cellular metabolic activity as an indicator of cell viability, proliferation, or cytotoxicity. So it doesn't need to lyse the cell like MTT assay. Neutral rate is more like a pickup of the neutral red dye of the viable cells. So these are different, I'll say different kind of concept because of different kind of dyes, but it serves as a purpose to showcase to you what is the cell viability. So the reason they pick these two because they want to have multi-dimensional results, so they won't miss out any important data, okay? Because of course they have their own drawbacks and perhaps certain I'll say these two livers that reacts differently to different kind of dyes. Okay, so that's why they included two types of assays over here. Of course, the measurement will be measured, absorbance will be measured at 400, 450 and 540 nanometer wavelength respectively, with a reference level wavelength at 690 nanometer wavelength. So this is data. So in this kind of publication, you will no longer see a very basic, I'll say representative image of the plate itself. You will see a data presented in a graph and in a transform format. So here we can see the plot over here where they showcase how these CYN transform product by the manganese oxidizing bacteria are less toxic to human liver cell. Okay, so let us look at this XDT. So it's easier that way. So between these two, I'll just pick this then. It's easier to look at it. So you have the control group, the initial CYN concentration and zero means no, okay, don't have. And you have at 1 and 3 milligram per liter. Then you can, as you can see, when there's no toxin in the media, the cells are viable. They are 100% viable. Okay, but when you introduce a 1 or 3, the viability drops a lot, drastically, where in a significant manner to, I'll say this is around 20 or even less than 10% of the cell viability. But if you look at the other group that have been that incubated with the transformed product, those transformed product are from the bacteria itself, the cell viabilities are basically almost the same as the as if there's no toxin at all. There's less toxin, okay, at different concentration. Okay, so it's just telling just a way to tell to plot the data and inform and present it to say that the toxicity if of the transfer product is indeed way lesser than after the bacteria churn up and transform the toxin. Okay, so it's very important because they will utilize this kind of information to proceed with, you know, the downstream study on how to utilize it on field. So as I said, Absorbance Microplate Reader is a very basic equipment, but it gives you such a very basic information so you can move on to the rest of your study. So whether you on how you want to I'll say analyze and also use the data and move forward. Of course, we have a lot more publications using Ao Absorbance Microplate Reader, so I will not go into details over here. But if you see something that sparks your interest and may be applicable to your field, please feel free to just crop this slide and then just search for the publication. Okay, so let's recap what we have talked about in today's session. So we talk about the basics of ELISA. We talk about the I'll say the capabilities or characteristic of the antibody, so you can enjoy the benefits of the ELISA kit and then get your data. The application that you can do with Ao Absorbance Microplate Reader is quite, I'll say, is a wide coverage. It's not limited to antibody. It can be done by any other dyes that give you or any other kit that give you colorimetric changes. Okay, and of course, when we look at the publication, you can also understand that it serves such an important piece of information, or not even mentioned in the publication itself, because it's such a basic equipment that everyone should have in their lab. Okay, so if you're interested, please feel free to go onto our website, azurebiosystems.com. We have all the information available on the site, with the features, application, and citation, and all the documents, and even video guides on how to use the Ao Absorbance Microplate Reader. Okay, right. Then, of course, there's a specification of the reader, but you can, if you're interested, please feel free to contact Apical Scientific so they can approach you and give you more information on that. So there's a few more citations we have, and we're Azure Biosystems. We have imaging solutions for every lab and solution, particularly for protein imaging. So we have Western blot reagents, Ao Absorbance Microplate Reader, Azure imaging systems that cover basic gel imaging, both chemi and fluorescence, Western blot imaging, and of course, we have the highest spec of all, Sapphire biomolecular imager, that you can do very advanced applications for fluorescence, phosphor imaging, and even Western blot imaging as well. So thank you for your time here today with us.